Okay, in our third part of chapter 8, we're going to look at actually how molecules, pigment molecules like chlorophyll, are able to absorb portions of the visible spectrum, those portions of light energy. So remember that as these molecules like chlorophyll absorb photons of light, they're also going to absorb energy. Now, they are not absorbing all of the photons of light energy because some of it is being reflected. Which wavelengths are being reflected by a chlorophyll molecule? The green wavelengths, right? That's why chlorophyll appears green. Now, if you think about this, when molecules like chlorophyll absorb the energy, these molecules of chlorophyll are going to be pushed to a higher energy level. If you absorb energy, you're going to be pushed to a higher energy level and you get excited. And we've talked a little bit during some of our lectures on energy conversions that as things increase their energy level, the stability is sometimes not maintained. So if a chlorophyll molecule absorbs portions of visible light, these photons of light energy, how can they regain or reach their stability? Well, one of the ways we've seen is for these molecules to lose electrons. And so, as a chlorophyll molecule, seen here, absorbs this photon of light energy, it goes up to this excited state. Now, when it loses this electron, it's going to also lose some energy in the form of heat, right? So excitation of an isolated chlorophyll molecule is going to cause this molecule to lose these electrons. And remember that when a molecule loses electrons, we say that it is oxidized. Now let's look at how this occurs on the chlorophyll's chloroplasts membranes. So here's our photon of light, and you can see here that as it's absorbed by this molecule of chlorophyll, right, the chlorophyll molecule gets excited. The energy level goes up. And so as the energy level goes up, as the chlorophyll molecule is excited, we're going to see the electrons leave. And remember that an electron acceptor becomes reduced in this process. Now, once we start moving these electrons, as you'll see, we're going to use them to drive some very familiar processes. But let's come back and see what's happened to our chlorophyll. As chlorophyll is oxidized to regain its energy stability, it's lost those electrons. So where are these electrons going to come from so that chlorophyll can be reused and allow another photon of light to strike it and cause the electrons to move again? Well, we're going to see these electrons donated, right? Now, think back to cellular respiration. The final electron acceptor in aerobic cellular respiration was oxygen as we formed a water molecule. As we'll see, we're going to pull apart a water molecule, removing some of these electrons that are going to be given to our molecules of chlorophyll. All right, so this slide here just reminds you that as photons of light energy strike our molecules of chlorophyll, they reach an excited state and they are oxidized as they lose their electrons. These electrons reduce an electron acceptor, and we'll talk more about them later. But to replenish these electrons, we're actually going to pull these electrons from water molecules. Now, photosynthesis is a two-step process. So the first portion of this process is called the light reactions. This is the photo portion of these reactions because it's going to require photons of light energy to drive them. The second part of our photosynthetic process is called the carbon fixation pathway. And it's here where we're going to take the carbon and fix it. To fix the carbon means that we're taking carbon in a non-usable form, like carbon dioxide, and we're going to rearrange them into a more usable form, like glucose. Sometimes this carbon fixation pathway is called the Calvin cycle, named for Melvin Calvin, who discovered this in the 1940s. It's also sometimes called the dark reaction. But I want you to learn it as the carbon fixation pathway, because naming it the dark reaction implies that it occurs at night, and that's certainly not true. The carbon fixation portion doesn't require light, but it does require the products of the light reactions, so that we are going to be able to fix our carbon and put the carbons into a usable form. Now, if we look at how these two portions of photosynthesis occur spatially in our chloroplast, as we'll see, the light reactions are going to occur on these thylakoid membranes, right? So remember, the thylakoids are those individual discs that make up the grana of our chloroplast. These thylakoids have protein complexes on them that we'll talk about in just a moment. And these protein complexes also contain the pigment molecules like chlorophyll. And so this is where the photo portion of our reaction occurs. As photons of light energy are absorbed, we're going to be using these to drive the light reaction processes. And again, notice at the very end, we have to replenish these electrons that we've lost. So we're going to use a water molecule to do that. And this, as we split apart this water molecule, we're going to liberate some oxygen. Now, the light-dependent reactions, or the Calvin cycle, or the carbon fixation pathway, however you want to learn it, occurs outside of the thylakoids in the stroma of the chloroplast. And it's here where we're going to take the products of the light reactions, which are some ATPs and an electron carrier molecule that we'll talk about in just a moment called NADPH, to drive them. The necessary energy is provided by the ATP and this electron carrier NADPH, and we're going to use this to fix our carbon. Our carbon dioxide will be converted into these organic molecules like sugar in these processes. So, two parts to photosynthesis: the light reactions and the Calvin cycle. Light reactions occurs on the thylakoid membranes. The Calvin cycle occurs in the stroma.